Hello everyone, my name is Aga and I'm a second year PhD student working at Queen Mary University of London and King's College London. I will talk about the meta-analysis of the role of genetics in neurodevelopmental disorders and their comorbidity with disruptive impulse control and conduct disorders during childhood and adolescence. A systematic understanding of the genetic and environmental etiology of neurodevelopmental disorders that we refer to as NDDs and of their comorbidities remains incomplete. A disproportionate number of studies, reviews, and synthesis of extant literature have focused on autism spectrum disorder, ASD, and attention deficit hyperactivity disorder, ADHD. However, other neurodevelopmental conditions, although showing similar prevalence rates and severity, have been overlooked. The aim of the current work was to meta-analyze the extant literature to first providing reliable estimates of the relative contribution of genetic and environmental factors to all NDDs. Second, to synthesize the literature on the etiology of the comorbidity between all NDDs. And third, providing an effective synthesis of the literature on the comorbidity between NDDs and disruptive impulse control and conduct disorders that we refer to as DICCs in childhood and adolescence. Studies for this meta-analysis were identified by searching Web of Science and OVID databases. A total of 12,432 studies were identified by two searches conducted 3 months apart. Screening and study selection was conducted by two independent reviewers and resulted in 324 eligible studies. Additional screenings resulted in 7 additional articles being identified, which resulted in a total of 331 studies and more than 4 million partly overlapping participants included in this meta-analysis. Our first aim was to synthesize the extant literature to obtain reliable estimates of the relative contribution of genetic factors to individual differences in all NDDs. When considering family-based designs in blue, we found that across all NDDs, the grant heritability estimate was 0.66. Grant heritability ranged from 0.86 for intellectual disabilities to 0.62 for specific learning disorders, but did not differ significantly across NDDs. DNA-based heritability estimates in orange were also not found to differ significantly across disorders, but could not be calculated for all NDDs. Our second and third aims were to meta-analyze the genetic correlations between different NDDs, as well as between NDDs and DICCs. This circular plot shows the strength of the meta-analytic genetic correlations, with each color indicating a different disorder. For example, the yellow line depicts the strength of the association between ADHD and conduct disorder. The thicker the color path, the stronger the genetic correlation between two disorders. Here we can see that the strongest genetic comorbidity across all NDDs was observed for ADHD and motor disorders, the genetic correlation of 0.90, and the weakest overlap between ADHD and specific learning disorders, 0.07. Overall, the genetic comorbidity was found to be as strong between different NDDs as between NDDs and DICCs. Genetic and environmental influences on NDDs were not found to differ by sex or by developmental stage, and effects were also highly consistent when we separated studies that had considered diagnosis and clinical cutoffs from studies that had quantified NDDs and continuous traits. We found that the genetic and environmental contributions to NDDs differed substantially as a function of geography, highlighting how estimates of genetic effects on disorders are highly sensitive to different environmental contexts. Our work on geographical differences also highlighted how research into causes of NDDs and their comorbidities is largely limited to Western countries. To conclude, this meta-analysis extends our knowledge of NDDs in several directions. First, it provides a holistic view of genetic and environmental contributions to all NDDs and to their comorbidities over development, revealing that NDDs are just as strongly genetically comorbid with other NDDs as they are with DICCs. Second, it identifies lack of neurodiversity in genetic research, which calls for future research to equally distribute the effort across all NDDs. Third, it equips clinicians and educators with new knowledge about patterns of etiological comorbidity between NDDs as well as between NDDs and DICCs, which we hope will inform clinical and educational diagnostics and practice, resulting, for example, in expanded diagnostic screening, that is, screening for NDDs accompanied by other genetically comorbid NDDs and DICCs, and the development of evidence-based interventions to improve classroom inclusion of pupils diagnosed with under-researched NDDs. Thank you so much for your attention.